name is Tess Leuthner, and I'm a PhD student in Joel Meyer's lab in the Toxicology and Environmental Health program at the Superfund Research Center at Duke University. The main focus of my research that I'm going to talk about today is investigating the long-term effects of persistent low-level exposures of pollution on DNA in various organisms. A healthy environment is intrinsic to our own health and ecosystem health. Myself and other scientists are interested in learning what fingerprints different chemical exposures can leave on DNA as a clue to better understand not only the identity of chemical exposures, but to try to understand the consequences on health of humans and other organisms. The sequence of DNA doesn't just code for different proteins, but it is responsible for all the biodiversity in our planet. Even within a species, such as this alpine wildflower, and even humans, changes in the sequence of DNA result in a lot of the variation that we can see between individuals. The source of this variation and diversity are called mutations, or changes in the sequence of DNA. For example, if we start with this sequence on the left, but in the next generation we see a T where the letter C had been, this is a C to T mutation. Not all the time, but sometimes this change in the sequence can change the protein or change what we see. Mutations happen randomly, but chemicals can also cause mutations to happen. This is because chemicals can cause damage to DNA. For example, the type of damage that ultraviolet rays have on DNA can sometimes cause mutations that result in skin cancer. And the polycyclic aromatic hydrocarbons found in cigarette smoke are known to cause DNA damage that can mutate DNA and result in lung cancer. But we are exposed to a lot of other chemicals every day, and we don't know how these chemicals can cause DNA damage and mutation. So myself and other scientists are trying to figure out, can we look at the sequences of DNA to understand if a chemical is causing a mutation? We hypothesize that just as each person's signature is unique in identifying who they are, mutational signatures may reveal what chemicals a person or organism has been exposed to. One of the chemicals that I study is cadmium. It is a pollutant found all over the world from human activities, such as industrial combustion and emissions, electronic waste, mining, drainage, runoff, and has even been detected in children's jewelry. Because of the many different sources of cadmium pollution, cadmium exposure is a significant concern because it can be found in air, water, and soil. Cadmium affects kidney function and other tissues, but it is also a known human carcinogen. So in the lab, I wanted to understand the effect of cadmium on DNA mutations. I study an organism called C. elegans, which is a microscopic organism that is used in many research labs, and importantly, it has its whole genome sequenced. In the lab, I raised C. elegans on normal plates for many generations. In parallel, I also raised C. elegans on plates that had a low level of cadmium, equivalent to the level that is measured in human blood in the United States. At the end of the experiment, I sequenced the DNA of the C. elegans, and then I counted the number and what type of mutation occurred in the DNA sequence in the cadmium-exposed C. elegans compared to the control. One result that I observed so far is that we see an increase in the frequency of the G to T or C to A mutation in the exposed conditions. And so we'll be doing more experiments in the future to figure out why this is occurring. In the meantime, I'm interested in linking experiments that I've done in the lab to polluted environments in order to understand the long-term consequences of pollution on the organisms that inhabit these contaminated ecosystems. One project I'm working on is using the freshwater species Daphnia to understand the effects of a century of cadmium pollution from industrial mining runoff in lakes in Canada. Another project is with killifish that inhabit the Elizabeth River Superfund site that have been exposed for decades to extremely high levels of polycyclic aromatic hydrocarbons. Our goal is that by investigating the DNA of these organisms, we will discover unique mutational signatures that allow us to better understand the exposures and the long-term consequences of pollution. So overall, if we can understand the fingerprints that chemicals leave on DNA, our goal will be to better understand environmental exposure with the ultimate goal of linking these exposures to various potential health impacts across communities and ecosystems.